Hi guys, in this video we are going to look in detail at the structure of the mitochondria, but what we are going to do is we are going to take a function first approach. The structures that it has specifically in relation to the function that it has to carry out. Okay, and hopefully looking at that, all its structures will make a little bit more sense. So let's begin. Aerobic respiration. So you will know from GCSE, respiration essentially is, as long as cells are supplied with glucose or have some glycogen that they can break down into glucose, as long as that's possible, you have glucose plus oxygen, and that oxygen indirectly is used to break that glucose down to give us carbon dioxide and water. So generally speaking, that's the reaction that's going to, or that the mitochondria contributes to. Okay, and in doing so, we release the energy that was locked in the glucose molecule, and we use that energy to make ATP. Okay, so this is the overall process. Now let's look at the individual process that result in the production of ATP from glucose. As well as that, we're looking at the consumption of oxygen. We're also looking for the production of carbon dioxide and the production of water. Okay, why this is important to the cell, we've already discussed in previous videos, that all the biosynthetic, there's so many processes in the cell that require the ATP. Okay, so let's put aside specialized cell functions for a second. But generally speaking, cells need ATP so that they can synthesize molecules, condensation reactions, any molecules where you go from simple molecules to complex molecules, small molecules to large molecules. Any of those kind of processes will need an input of energy. So ATP is used for that. Any situation where the cell needs to carry out transport of molecules against concentration gradients, for example, obtaining molecules at a low concentration outside of the cell, importing those molecules into the cell so that it can have an energy store, for example, so that it's got biomolecules to use in synthesis. All of that transport against concentration gradients will require ATP. So, and we also discussed that membrane fusion requires ATP, membrane deformation, the formation of vesicles that allow materials to transfer from one organelle to another, that all requires membrane deformations that are energy requiring processes. So there's a lot of processes in the cell that require ATP, not to mention the specialized cells such as muscle cells that need ATP for muscle contraction also. So ATP is really essential for the cells, and that, generally speaking, is where the mitochondria comes in. Let's not forget that cells can generate ATP anaerobically. Okay, so in the cytoplasm, the glucose, which I'm going to simplify to G now, the glucose can be converted to lactic acid, and this lactic acid can be produced and then it's metabolized, and as a consequence of that we can get some ATP made, but it's a very small amount. This is the key point, that anaerobic respiration, i.e. respiration without the mitochondria, anaerobic respiration can generate some ATP, but aerobic respiration, so whereas anaerobic respiration for each glucose gives us, say, 2, aerobic respiration, i.e. plus mitochondria, aerobic respiration gives us around 30. The numbers can vary, but it's around 30. Okay, so we get 15 times more ATP because of the presence of mitochondria. So aerobic respiration is therefore a very important adaptation of certain cells because it allows them to extract more energy from their energy or food molecules. Okay, so their energy stores can be more productive by the use of aerobic respiration. So the presence of mitochondria is very, very important. Okay, so before we go too much further, let's get some basic mitochondria navigation done. What we have here is the outer mitochondrial membrane. Okay, so I will abbreviate that to outer mitochondrial membrane. Then we have the intermembrane space. So the space between the inner membrane and the outer membrane is the intermembrane space. Next, shall I move here? So next we have the inner mitochondrial membrane. So that would be this one right here. So we have the inner mitochondrial membrane. Then moving, actually sticking with the inner mitochondrial membrane, a special part of the inner mitochondrial membrane, actually I'm going to stay up this side. A special part of the inner mitochondrial membrane is these infoldings that increase the surface area called cristae. Okay, so I'll go this way. Okay, so it is a part of the inner mitochondrial membrane, but it is a specialist part of the cristae, as we'll see when we will look more closely at the structure. Okay, so we have a specialist part of the inner mitochondrial membrane, the crista, and then we have that fluidic region in the middle or in the center, enclosed by the inner mitochondrial membrane, which is the matrix, or the mitochondrial matrix. Okay, now within that matrix we have some circular DNA. Now these things wouldn't be visible, so what I've labeled here, that might be visible under an electron microscope or in an electron microscope image. The following will not be visible in an electron microscope image. As far as I know, that's the circular DNA that has some genes that are used by the mitochondria, and if you got genetic information, the way you convert that genetic information into useful product, protein or protein product, is because you have enzymes such as RNA polymerase that's going to copy the genes into mRNA, and you're going to have ribosomes that are going to use the mRNA to build proteins for you. So the way that the mitochondria makes use of that genetic information is the ribosomes and some other enzymes. Okay, so the matrix is full of enzymes, as we'll see. Okay, but that's the basic navigation of this structure that we're looking at. Now we'll start looking at the function of the mitochondria as a way to look at the detailed structure of the mitochondria. All right, guys, so let's continue. So aerobic respiration has 4 stages. The first of which happens in the cytoplasm, but it's only when this first stage happens that the second stage can happen, so we need to look at it. Okay, and the first stage is called glycolysis, where glucose is broken down or oxidized to the molecule pyruvate. Now the important thing for us is that glucose is a 6-carbon molecule, whereas pyruvate is a 3-carbon molecule. So because we've caused some breakdown, we've broken that molecule down a little bit, energy is going to be released. We've gone from complex to simple, energy is released, and so that has allowed us to make or convert some ADP into ATP, but also we've extracted some hydrogens and from the glucose, and we've transferred them onto a hydrogen carrier molecule called NAD. Okay, it's a coenzyme and it's carrying hydrogens for use later on in the process. So you've got to keep your eyes on ATP and you've got to keep your eyes on NAD. So once the NAD is carrying hydrogens, we call it NADH or reduced NAD. Anyway, The point is that we have now made pyruvate, and pyruvate is going to enter the mitochondria, and the aerobic stages of respiration will begin. This stage is called glycolysis. So stage 1 is called glycolysis. Okay, now pyruvate then enters the mitochondria. So pyruvate has to go through the outer membrane, through the intermembrane space, and through the inner mitochondrial membrane and into the matrix. Okay, so there it is, and so it now is here, okay, in the matrix. Now, in the matrix is where the next stage happens. So pyruvate is next converted to acetyl coenzyme A, or it's added to coenzyme A. So coenzyme A comes along and the pyruvate is converted, so the 3-carbon pyruvate is converted to 2-carbon acetyl-CoA, and we've lost some CO2 there. Okay, so we've produced some CO2 now, carbon dioxide. Once we've got acetyl-CoA, actually, and we've also produced some NAD. H. Okay, so this is important. We've made some NADH here. We have made some NADH here. This is called the link reaction. Okay, now what I'm thinking is how I'm going to use the available space to do the next part. But anyway, so that's the link reaction. So that's stage 2, to the link reaction. Trust me, this is all going to have connections to the discussion of structure when we get to it. Okay, so thank you for bearing with me. Next, what happens to the acetyl-CoA? Now the acetyl-CoA next goes into the third stage, and the third stage is called the Krebs cycle, or the tricarboxylic acid cycle, TCA cycle. Okay, so stage 3, Krebs cycle. I'm going to cut a long story short here for obvious reasons, but basically the acetyl unit goes into a cyclical metabolic pathway, the overall job of which is to break down further the 2 carbons that are still in that acetyl unit. Remember, we went from 6 carbons to 2 times 3 carbons, 2 pyruvates right there. We've broken it down. Now those 3 carbon molecules go in here. They become 2 carbon molecules, and now the 2 carbon molecules are going to get further broken down. We are going to get further release of CO2. We are going to get some ATP production. So some ADP is going to be converted to ATP here. But the main thing that happens here is the generation of reduced coenzyme. So more NAD is converted, so more hydrogen is extracted and converted to NADH here. Okay, so as I said, keep your eyes on the NAD. Right, so we made some here, we made some there, and we've made some more here. Why? What's so important about NAD? Let's get to that in stage 4. So, can I move down there? I think I can. Okay, so now the NAD is going to get used in stage 4. Now stage 4 is called oxidative phosphorylation. It might also be called electron transport. It might also be referred to as chemiosmosis, but it's all happening on the inner mitochondrial membrane, and it uses now all this reduced coenzyme that we've made, NADH, is going to be used in this last stage. Okay, so what happens is, I'm going to get rid of this DNA for a second. Okay, don't worry, you can still produce. I'll just move it out of the way. Okay, right. Right, so now at this point the NAD is going to be used, and the inner mitochondrial membrane, the crystal part of the inner mitochondrial membrane, is very important in stage 4 because there's electron carriers. Now again, I'm going to get rid of a lot of detail here, but there are electron carrier protein complexes here. Electron carrier protein complexes, and I'm omitting some detail. Okay, as well as very important protein called ATP synthase in the inner mitochondrial membrane here, and we'll see what they do. So what happens is that this NAD comes down here, or the NADH, and the NADH delivers all the hydrogen. So all this NADH that was produced arrives now here at this electron carrier, and we'll call it the electron transport chain. Okay, and the NADH delivers its electrons. So the hydrogen at this point splits into electrons and protons. The protons are pumped into the intermembrane space. Okay, the protons are pumped into the intermembrane space, but it's the energy of the electrons that is used to pump the protons against a concentration gradient into a region where there is already a high concentration of them. Okay, so the NAD comes out the other side without its hydrogen. Okay, so as the electron flows through, so the electron is called the electron transport chain because the electrons move from one carrier to the other. As the electron moves through from one electron carrier to another, so the electron moves from there to there, first carrier to second carrier, the second carrier can pump another proton into this region. The electron then moves to the third carrier, and the third carrier can move a proton into the intermembrane space against the concentration gradient, building up the H+ concentration in the intermembrane space. Okay, and this is all thanks to the hydrogens that were carried or collected at each of the stages preceding. Now, there's a huge H+ concentration here now, and a much lower H+ concentration in the matrix because they've been pumped into the intermembrane space using the energy of the electrons. Now, the protons are allowed to diffuse back into the matrix through the ATP synthase complex. But as they move down their concentration gradient, and remember, when anything happens passively, it releases energy out into the universe. Okay, so when something moves down its concentration gradient, it releases a little bit of energy, and as these protons move through the ATP synthase complex from the intermembrane space into the matrix, they release energy, and the ATP synthase uses that energy to convert ADP into ATP. Okay, and it does that, or it can generate 30 ATP molecules for each glucose that took part in glycolysis. So there we have it. That's our fourth stage. That stage is called oxidative phosphorylation because we are using the energy of we've oxidized that electron carrier. So because we extracted the hydrogen away from that electron carrier, we've oxidized it, and that is what was used ultimately to form and phosphorylate the ADP to ATP. So oxidative phosphorylation, that is stage 4. So 4 oxidative So stage 4 was oxidative phosphorylation. Remember that we have used up some glucose, we have produced some carbon dioxide, but we have not used oxygen yet, and we have not produced water. That happens at this last electron carrier. So the electrons have to flow into something. They cannot just occupy permanently the spaces within the electron carriers. So what happens is that oxygen comes along. So this is why the mitochondria need, or this is why cells need oxygen. This is why it's called aerobic respiration, because the oxygen comes along and collects the electrons. So what happens is that the electrons from the final electron carrier, they join with a proton and with oxygen to form water. Okay, so one electron plus proton gives you hydrogen. This happens twice to give you 2 hydrogens, and it combines with 1 oxygen to give you a water molecule, H2O. Okay, and so for each molecule of oxygen that comes in, 2 water molecules would be produced. Okay, so here, those are the 4 stages. Now what we'll do is we'll look at how the structure of the mitochondria is adapted to allow all that stuff to happen and allow it to happen as efficiently as possible. Outer mitochondrial membrane. How is it that the outer mitochondrial membrane helps the process of ATP synthesis to occur? So primarily, it's not just there for no reason, nothing is. Primarily, the outer mitochondrial membrane is there to help proteins that the mitochondria need to be in the mitochondria. Okay, I know we've referred to this circular DNA of the mitochondria, but for most of the proteins, so all these enzymes that are needed to carry out the link reaction, the enzymes that are carrying out the reactions of the Krebs cycle, the protein complexes that are in the inner mitochondrial membrane, not all of these come only from the genes in the circular DNA. Many proteins that the mitochondria requires to carry out these processes need to be imported because they were produced using genes in the nucleus. This is how close the symbiosis between this prokaryotic structure and the cell in which it resides, so close has become that relationship that it can't even produce all the genes it needs to survive anymore. So those genes in the nucleus were transcribed, they were produced on ribosomes, and now those proteins, those polypeptides, need to be brought in here so that they can do their job. What we'll do is indicate that there are enzymes that are carrying out these processes. You know, this is a protein, these are proteins. Okay, so how do these proteins that are not synthesized within the mitochondria, how do they get into the mitochondria? And that is due to specific proteins that exist within the membranes of the mitochondria. So what happens is that polypeptides are synthesized in the cytosol or cytoplasm, and these proteins then allow these polypeptides to be pulled in to the mitochondria. So this process obviously begins in the outer mitochondrial membrane, and therefore it has specific proteins that allow the import of polypeptides from the cytoplasm into the mitochondria. Now, the inner mitochondrial membrane has to have proteins that complement this process and allow it to be completed, unless that protein works somewhere in the intermembrane space. But for proteins that need to end up within the matrix, for example, these polypeptides continue to snake inwards, and then once they're in the matrix, they adopt their final tertiary structure and start doing their job. Okay, so alternative processes might result in these in polypeptides forming their final tertiary structure in the inner mitochondrial membrane, and then making their way into the crista, where they eventually do their job of being an electron carrier or some other enzyme, or ATP synthase, for example. Okay, so if we're looking for outer membrane proteins, and the outer mitochondrial membrane has got specific proteins for polypeptide import. So that's its particular job. But otherwise, it also has to allow the free movement of things that the mitochondria will need to carry out its role. So it's got pores, protein pores. So I'm going to draw them like this. Okay, so these pores are in the membrane, and they allow the free diffusion without too much hindrance to molecules such as pyruvate, to other molecules that the mitochondria might need. Okay, so we're going to focus on the specific function of aerobic respiration. So pores in the outer mitochondrial membrane allow the diffusion in of pyruvate. Also, it's not only glucose that is used for respiration. Okay, so it's not only glucose, but also fatty acids. Fatty acids are an important respiratory fuel. Now, fatty acids also, so fatty acids will have other mechanisms by which it enters into the mitochondria, because it's hydrophobic in nature. So it's going to have a different mechanism. But there's specific proteins in the outer mitochondrial membrane that will allow the entry of fatty acids into the mitochondria, because it will be important for fatty acids to also be eventually converted into acetyl-CoA, and for acetyl-CoA that was made using fatty acids to then enter aerobic respiration, just like carbons from glucose. Okay, so outer mitochondrial membrane, key points about its structure are: it's got specific proteins for polypeptide import. It has got porins for entry of small molecules and ions. And what else? It has carrier proteins for import of fatty acids. Okay, so these are key things necessary for the function of the mitochondria. Okay, so that's the outer mitochondrial membrane. Let's move on now to the inner mitochondrial membrane. Complementary to that, the inner mitochondrial membrane, as we've discussed, it's much more selective. Okay, so it's much more selective than the outer mitochondrial membrane. Because the outer mitochondrial membrane has got these pores are much less selective. So things like water, amino acids, small molecules, mineral ions, they can freely diffuse in and out of these pores. Whereas the inner mitochondrial membrane is much, much more selective. So the inner mitochondrial membrane has got specific proteins that are selectively allowing things to move in from the intermembrane space to the matrix in a much more selective way than things move from the cytoplasm into the intermembrane space. Inner mitochondrial membrane is more selective. So the key structural features about the inner mitochondrial membrane is that there are much fewer pores. That's what makes it more selective. So it's got fewer pores, and it's got specific, so to selectively import, export, or selectively allow movement across that inner mitochondrial membrane, there's more specific transport / carrier proteins. Okay, so there's more of these specific carrier proteins that are specifically allowing the movement. So for example, pyruvate entered the outer mitochondrial membrane through a pore, but it enters into the matrix from the intermembrane space through a very specific protein carrier that only, or very selectively, allows the diffusion of pyruvate from the intermembrane space into the matrix. Okay, so it's got specialized proteins. So the protein content of the inner membrane is going to be very different to the outer membrane. Okay, similarly, there'll be specific proteins for the entry of fatty acids into the matrix, so that the fatty acids can be used to produce acetyl-CoA, as well as pyruvate being used to make acetyl-CoA. Okay, so that's the inner mitochondrial. As well as that, remember, there are going to be proteins that are involved in the import of polypeptide chains into the matrix, because obviously they'll have to move through the inner mitochondrial membrane as well to get into the matrix and work. So proteins, again, for import of polypeptides. Okay, so that's the inner mitochondrial membrane. Its specific structural features, obviously there's a lot more to it than we can discuss here, but we're trying to keep our focus on respiration and what are the structural features that allow respiration to continue. So in order for that, we need fatty acids to come in, we need pyruvate to come in, and we need enzymes to be working and proteins to be working in the mitochondria. So those two membranes have specific roles in that. Okay, we'll come back to the inner mitochondrial membrane, but next let's look at the matrix. So once pyruvate and fatty acids are in the matrix, what we need are enzymes to carry out the reactions to produce acetyl-CoA, and then we need enzymes of the Krebs cycle to carry out those reactions and produce NADH. Now, we've already discussed that the way those enzymes got there was through the outer and inner membrane protein complexes that allow the import of these polypeptides and for these enzymes to exist. So the specific adaptations, as it were, of the matrix would be that it has proteins and enzymes for the two stages of respiration that occur there, i.e. link reaction. So for the link reaction and Krebs cycle. Okay, so that's number one. Second is that the matrix is the container, or the matrix is the location of the circular DNA. So it contains the circular DNA. Okay, that's going to have the genes or some mitochondrial genes. Okay, so the circular DNA, but also, as well as the circular DNA, there's also going to be other enzymes needed to express those genes. Things like RNA polymerase, things like ribosomes. Okay, a special note on the ribosomes, however, they are the prokaryote style ribosomes. So slightly smaller than the eukaryotic ribosomes that might exist in the cytoplasm. So just something to bear in mind there. Okay, so that's our matrix. Okay, now finally we have the crista. So the crista is a part, technically it's a part of the inner mitochondrial membrane, but the protein content of the crista is very different from the part of the inner mitochondrial membrane that is in this part, for example. So here you might have lots of transport proteins, but the crista are specifically devoted to oxidative phosphorylation, that fourth stage. Okay, so the specific adaptations or structural features of the crista is going to be the folding. So the folding causes a high surface area, right? So all these folds, so the crista infolding of the inner mitochondrial membrane increases the surface area. Okay, that means it can contain more, right? It can contain more electron carriers and ATP synthase. So these particles here, okay, ATP synthase for oxidative. So for the oxidative phosphorylation stage of a respiration. So yeah, the more folding, the higher the surface area, the more of these components can be contained here, and the more of these components that are there, the more efficiently it can make use of NADH provided by the Krebs cycle, and we can more efficiently generate ATP. Okay, so is there anything else? Right, for the crista, just want to mention the ATP synthase. In certain electron micrographs you can see these. So these, so this part of the ATP synthase is visible. So not under light microscope, but in certain electron microscope images, they are visible as stalked particles. So maybe just something to be aware of there. Okay, and is that it? I think that is it. So there we have the key structural features of the mitochondria that allow the mitochondria to function, to carry out respiration. Okay, so this was our look at mitochondria, essentially their structure and their function, or how the structure is related to function. Next time we'll look at chloroplasts in a similar way.